Now let's discuss the pond width, the procedure. We place a pond width, example hydrilla, upside in a test tube containing sodium hydrogen carbonate solution. So if the test tube is this way, we cut the pond width with the bottom facing upwards and then the leaves facing downwards, because we are going to turn this test tube upside down. So once we turn it upside down, the pond width will now look this way, the bottom here and then the leaves pointing upwards. Okay, now we place the test tube in a beaker of fresh water at 25 degrees Celsius. At 25 degrees Celsius. So that is why we need our thermometer, so that we will use that to measure the temperature all the time. The third thing we do is that we darken the room. You're going to darken the room. This is because you want the light source to be the lamp only, or we want the lamp to be our only light source. Now we place the lamp or the light source at a fixed distance from the plant. Now this time around we are going to test the concentration of carbon dioxide. So the distance from, or the distance between the lamp and then the plant will not be varied. We are going to place it at a fixed distance. Next, we are going to count the number of oxygen bubbles given off by the plant in a 1 minute period. Our stopwatch comes in here. This is a sign of the rate of photosynthesis at that particular concentration of carbon dioxide by using different saturations of sodium hydrogen carbonate. Okay, so we're going to measure the amount of the amount of oxygen bubbles that are going to be produced in a 1 minute period. Now we are going to repeat the experiment for different concentrations of carbon dioxide and count the number of bubbles produced in each case. Now in sodium we are going to use different saturations of sodium hydrogen carbonate solution. We'll graph the results and then plot the concentration of carbon dioxide on the x-axis, because as the concentration of carbon dioxide increases, the photosynthesis increases, the rate of photosynthesis increases. So the concentration of carbon dioxide is an independent factor. It is a factor that can stand on its own, but the rate of photosynthesis depends on the concentration of carbon dioxide. That is why it is on the y-axis. In the end, we are going to test to see if the gas produced is oxygen. We test oxygen by seeing if it will relight a glowing splint, and then we make sure we keep the temperature of the room at 20 degrees Celsius.